is Daniela Zarate, who is a postdoctoral fellow at UC San Diego as well as UC Riverside. And the title of their talk is Admixture in Africanized Honeybees, Apis mellifera, from Panama to San Diego, California.
Yay, awesome. Super happy to be here. Thank you so much for the introduction, and let's get to it. This has been a really amazing conference, so thank you so much to all the organizers. And I'm going to be talking about my dissertation work that I did at UC San Diego. I'm currently at UC Riverside as a postdoc with Jessica Purcell, studying supergene evolution in Formica, but I haven't forgotten my honeybees, and I still want to continue to do work in honeybees. But I'm going to be talking about what I learned concerning genomic admixture in Africanized honeybees across the American continents. First of all, I want to say that I'm a general biologist interested in genetics and behavior of social insects, and which is why I moved on to work in ants from bees. I think it's been such an amazing journey working with bees to ants. And broadly, I would say I was really interested in this idea of admixture, hybridization, genomic mixture of genetically distinct lineages, which kicked off this project. And as we all know, honeybees like to mix and mingle across subspecies. So they're a perfect study system to study admixture and how admixture shapes genetics and behavior. And I was particularly interested in the Africanized honeybee. As we all probably know, honeybees aren't native to the Americas. They're introduced species worldwide. They're divided into these 4 major biogeographic clades: the African, Western European, Eastern European, Middle Eastern, and also we have additional clades that are continuing to emerge as new science is done. And honeybee phylogeny spans across 30 subspecies, and I just have a small representation of that subspecies diversity here in this phylogeny published by Webster. But I was particularly interested in a hybrid honeybee, the Africanized honeybee. So brief refresher, Africanized honeybees. The story really starts with European honeybees introduced to the Americas as early as 1622, various mellifera lineages. And it wasn't until 1957 that Scutellata, the sub-Saharan widespread African honeybee, was introduced and spread across the continents. The current northern range limit is as high north as Napa and Sacramento County, San Francisco area, in California. So it really has become quite an American phenomena. And this bee has been characterized as super defensive. It's been given the name killer bee by the popular media because it's so defensive. And so I was particularly interested in studying nuclear admixture and mitochondrial ancestry, as well as genetic diversity in Africanized honeybees across its range. And there had been work done in southwestern United States and in Brazil, and to some extent in Mexico, but whole genome work there only had been a few studies that had done that. And so I was interested in just expanding what was already done and including more sites in understanding admixture in Africanized honeybees. So the first thing that I did was made good use of 2 available sequencing projects, Wahlberg and Harper's sequencing projects that we had sequenced up various subspecies of mellifera, and so I collected 99 from NCBI. And so I had my reference panel built from these sequencing projects, and I collected 60 Africanized honeybee samples in 4 locations in San Diego, because I'm in San Diego. I collected in Chiapas, Mexico, Santa Rosa National Park in Costa Rica, and in Panama City in Panama. Largely this selection, the sampling scheme was largely driven by our collaborators and who we were able to tap into to be able to sample. So it was just 4 locations, but we felt it was like a good little start to studying admixture. And for getting to that first question of how does global ancestry vary in Africanized honeybees, I basically chopped off their heads and extracted DNA, sequenced to relatively high coverage, and did some pretty standard bioinformatic quality control, both for nuclear DNA and mitochondrial DNA. And I'm just glossing over the details here. All the nitty-gritty is in our paper that was published this year. So definitely feel free to check that out if you want to get more into the details. And so just general bioinformatic calculation of genotype likelihoods, we ended up using 2 million SNPs to do a principal components analysis. And for the admixture itself, we used about 320,000. So to start off with, there was a strong signature of population structure with the PCA. So in PCA space, the Western European, typo, that should be Eastern European, the Middle Eastern and the African clades all kind of fell on around the PCA space, bunching together as expected. And then the admixed populations grouped pretty interestingly. The Mexico, Costa Rica, and Panamanian populations grouped relatively closely to each other and closest to the African genomes. And then our San Diego population kind of fell in the middle of all of that PCA space. And it kind of made sense when you look at the actual admixture itself. So this is an admixture plot. Each column is an individual honeybee, and the colors correspond to the ancestry from the 4 different clades. So first we have our old world honeybee reference panel, the bees sequenced by Harper and Wahlberg previously, and I'm just showing a little piece of it here. These are the 21 Scutellata honeybees that I included, and as you can see, they're all pretty red, so they're all pretty 100% African. The Western European, pretty black bees there, so we have a little bit evidence of admixture in one or a couple individuals, but largely they are mostly Western European in ancestry. Eastern European, same story. Middle Eastern, mostly groups by itself, but there is some evidence of admixture there in the Syrian population. But moving on to my first sampled population, San Diego, California, it's highly admixed. About 38% of that ancestry is coming from the African. Chiapas, Mexico, we have about 77% on average coming from the African, and then Costa Rica and Panama, that admixture proportion increases. So that was really interesting. And then I grabbed the mitochondrial DNA, and I just also downloaded some representative mitochondrial DNA from NCBI and aligned it, created a phylogeny, and this is it. But before you squint at it, I made some bar graphs and some tables for a better visualization. But basically, mitochondrial ancestry, by and far, I would say reflects kind of the nuclear admixture analysis. We have a lot of African mitochondria. 100% of the population in Costa Rica and Panama has African mitochondria, with a little bit less in San Diego and Mexico. And again, in San Diego, we see a little bit more representation from other honeybee clades. And then my last question was, how does genetic diversity vary across Africanized honeybee populations? To calculate this, we did a genome-wide pairwise estimator and the genome-wide Watterson estimator, two just different types of calculations. And then also, in order to control for the fact that Wahlberg's bees were sequenced to a lower depth than both mine and Harper's sequencing set, we just calculated genomic diversity using high quality SNPs. And when we did that calculation, we weighted it by the number of sites in the genome. And also the other two genome-wide estimates, I calculated standard error by block jackknife technique by removing each chromosome at a time. And so this is the first results of that. And on the top is the bar graphs. The first 4 bars are the pure honeybee populations. And then the 4 bars on the right with the magenta colors are my admixed Africanized populations, and the table below shows the numbers and the standard error, and the lighter the numbers, the lower the genetic diversity, and the darker that mustardy color is, the higher the genetic diversity. So what pops out immediately is the African population is highly diverse, the European and the Middle Eastern, not so much, and then our admixed populations are as diverse as the pure African clade. And that was, we replicated that very similar pattern if you use the pairwise estimator just using the high quality SNPs. And when you use the genome-wide Watterson estimator, it's still pretty similar, but the African population is much more diverse than the admixed populations. But by and large, I would say this is pretty telling the same story of high admixture and admixed honeybees, and high admixture in the African, and low diversity in the European clades. So in conclusion, in terms of admixture, this pattern emerged of African ancestry does decrease with increasing latitude if you look at our 4 populations in space, and that San Diego bees are highly admixed, which is super cool because I'm a born and bred San Diegan, very proud of my city, and I like to see how diverse our honeybees are there. And how will mitochondrial ancestry differ and reflect nuclear ancestry admixture? I would say the mitochondria roughly reflected the nuclear. If you noticed, we actually didn't find any Eastern European or clade C type mitochondria in our San Diego sample, but honestly, I think that's just an artifact from how small our sampling site was. We only had 15 honeybees, and a previous mitochondrial study that sampled much more broadly, which actually my PI published, showed that they did find Eastern European mitochondria in San Diego. So I don't think it's because there isn't any. Lastly, genetic diversity. Africanized honeybee populations have high levels of genetic diversity, which was cool, not super unexpected, but what was unexpected was that the San Diegan population was as high of genetic diversity as the Costa Rica, Mexican, Panamanian, because as we had known previously, African honeybees are more diverse than the European or the Middle Eastern, and so I would kind of expected like a honeybees of high African ancestry, like what you see in Panama and Costa Rica, which is 90%, 85%. It would have high genetic diversity because it has so much African ancestry. But the San Diego population only has 40% African ancestry, and it's as diverse as an admixed population that has 90% African ancestry. So that was kind of cool. And future work that I want to do a local ancestry analysis and actually map out which parts of the genome are coming from the African versus the European. And also coupling this admixture data with behavioral data and nest defense, which is something I'm trying to get published right now. Is I did a really cool project assessing nest defense in Africanized honeybees. I've gotten stung hundreds of times at this point in my life and just have to see it published at this point. And the last thing I want to leave you all with is this idea of, should we rethink the name Africanized honeybee? Because when I first started grad school, I just called it Africanized honeybee. Everyone called it Africanized honeybee. And the more I studied it and the more I became familiar with broad, honeybee diversity, I became a little bit more convinced that we should move away from this Africanized honeybee term, because as I'm sure most of you know, African honeybees comprise several subspecies that vary in defensiveness and other behavioral aspects. There are more than 24 African honeybees, and so to kind of pigeonhole just one subspecies as the Africanized honeybee seems to me a disservice. And also we kind of now have associated like the term Africanized with high defensiveness, invasiveness, pest type of honeybee. And there are African honeybees out there that are surprisingly, air quotes, gentle. And so like the Ethiopian honeybee, mellifera monticola, has been noted for its gentle behavior. And so I think it'd be cool to kind of start changing the name surrounding this hybrid honeybee. My personal favorite, I would like to call it the American honeybee instead of the Africanized honeybee. I think that would be pretty cool. And I just want to thank a whole bunch of people that helped me with this project. And again, if you're interested in the nitty-gritty of this project, we did publish this in Ecology and Evolution this year. So yeah, I'm happy to take any questions at this point.
So we're just at 3:30, so I don't think we can answer questions, but there is a question for you in the Q&A, and I think you might be able to type into it if you want to check it out. And if anyone does have questions, just, you know, DM them to Daniela, and I'm sure she'll be happy to answer them.
Awesome. Thank you so much, everyone.
Yeah. So the